we view this as a very important form of outreach where we can share with you the excitement of what inspires us and excites us. Many of you tonight have been with us from the start, last year and this year's series, and as you know, this year's theme was to feature distinguished faculty members in the Department of Ecology and Evolutionary Biology. We're extremely grateful for those of you who helped support this series because it does allow us the opportunity to share our excitement of our faculty and our research with a larger audience, and certainly beyond the walls of our lecture halls. And we hope to continue to bring you exciting and inspiring research from our department and from other places in the coming years. It's really never been more important to understand the diversity of life, and our department, Department of Ecology and Evolutionary Biology, really provide a vital role in discovering nature. Now, it's my special honor and pleasure tonight to introduce my colleague, Robert Wayne, as tonight's Darwin Evolving lecturer. Bob's a professor in the Department of Ecology and Evolutionary Biology. He's a renowned molecular geneticist and evolutionary biologist, and he's truly one of UCLA's distinguished naturalists. Bob received his PhD from the Johns Hopkins University and later was a research biologist and postdoctoral investigator for the National Cancer Institute. He's the former head of conservation genetics at the Zoological Society of London. He's authored over 180 publications, and his work has always been on the cutting edge of molecular genetics, evolution, and conservation. He's a world authority on the genetics of dogs and wolves and their relatives, and he's going to be sharing with you some of that work tonight. He started here at UCLA in 1985, first as a research associate, and for the past 12 years has been a professor. In addition to being a professor at UCLA, Bob's an editor, or is current or past editor for a number of high-profile international journals like Molecular Ecology. He does a lot of applied research and is a member of the IUCN Captive Breeding and Canid Specialty Groups, and he's a research associate for the LA County Museum of Natural History. So I hope tonight you're going to enjoy learning more about Bob's fascinating work as much as we do. Please join me in welcoming Professor Robert Wayne, who will share with us the evolutionary history of the domestic dog.
Well, thank you, Dan, and good evening, everyone. If we could just dim the lights a little bit so we can see the slides better. Can we do that? Do I control that, do you think? I'm going to try to do it myself. There we go. Save the applause to the end. Well, I have to say it's truly a wonderful time to be an evolutionary biologist who's interested in genetics. Just looking back at my own history, when I got my PhD in 1984, one could have an entire PhD that was based on maybe just a handful of DNA sequences, maybe comprising a few thousand base pairs. Nowadays, in 10 days, we can run a certain machine that will produce over 120 billion base pairs of information, and that then can become just a small part of a much larger PhD thesis. And for me, the world has been turned over in a sense. It's changed so dramatically. The last 2 decades, before 2000, for me, were dominated by population studies that maybe involved a handful of genes, maybe 6 or 10 markers, and from that we try to make inferences about what's going on in the entire genome and in the history of the species. Now, we contemplate studies of all the genes in a population and how we're even going to handle that information. But it's the difference between going into a field and plucking a few flowers and making generalizations about what's in that field, to actually sampling every plant and every microorganism in that field, and then putting that together to understand the community ecology of that field. We're now at that point in population genetics, sometimes now called population genomics, because we're looking at the entire genome rather than just a subset of genes. Well, you can imagine that that kind of information is really dramatically revising our notions and testing ideas, long-standing ones, and many of them are being turned over. And what I want to give you an idea of is some of these ideas, how they've changed in understanding the domestication of dogs, and their relationship to their wild kin. You know, I'm just going to change this a little bit here. Gonna come back though. Yeah, if you could turn the lights back down. We're at opposite purposes here. Okay, so just by way of introduction, I'm going to start off my talk and telling you a little bit about why I like dogs, and it's not the reason you think. You know, sure they're great and look you in the face, but actually that's not the reason why I started working on dogs. In fact, I don't even own a dog today. I own 2 cats. I'm then going to kind of lead into a bit of an evolutionary framework, because this is where Darwin started. One had to view the evolution of dogs in an evolutionary framework with relationship to the entire family, Canidae. And then we'll talk directly about dogs, their origin and diversification, what we knew until about a year ago, and then specifically the new data that has come from sequencing of the dog. And these discussions of the new data will be broken into several parts. First, just about how we build a dog. You know, what we know actually about how we create new dog breeds, and of which there are hundreds. And I'll talk about some specific genes that we've been able to find that influence conformation and form of a domestic dog. And this has been a very dramatic discovery for us, because previously these genes were not known. And I'll have this kind of general discussion of the number of genes that make a dog or dog breed different from other dog breeds. And then kind of end the data discussion with trying to bring it all together and synthesize everything and talk about how we actually evolve a dog. I then want to talk about a very unusual finding that we also discovered within the last year, and that is how genes that have evolved in dogs have been transferred to wolves and have become successful, at least in North American populations of wolves. And this is something, you know, we talk about GMO foods and genetically engineered this and that, but we haven't really talked about how products of domestication can not only establish themselves in the wild, but actually transform an entire population. And lastly, I'll talk about what we're doing now at UCLA and the Wolf Genome Project. This will be potentially, if we get it going, the first genome, non-human genome, ever sequenced at UCLA. All right, a little bit about why I like dogs. You know, of course, they're adorable and our best friends in some respect, but why I like them is because I was interested in adaptive radiation. That is, the rapid production of forms in the fossil record, the Cambrian explosion, the radiation of birds and mammals, of fish, such as cichlids in the Rift Valley lakes in Africa. How one can explain, from a developmental and genetic perspective, this rapid diversification of form which is adaptive. And dogs seem to me to be a good model of this process, and that they show such a remarkable diversity in form and function. If we can explain some of the rules about how this kind of diversification occurs, then maybe we can understand how such diversifications occur in the wild. And where I started in the early 1980s, because this is before, you know, PCR and before really modern molecular techniques, I started with morphology. And that obviously was what has changed in domestic dogs. And I was very fascinated by this kind of change in skull shape and limb proportions. How could that have occurred so quickly? And there are certain rules to how dogs diversify. Many dogs are kind of have this juvenile or puppy-like conformation, big globular skulls, very short, tucked-in faces, whereas many adults of larger breeds have the more wolfy conformation. And my approach to this was actually started by studying the development of dogs. And I even radiographed breeds of different sizes and proportion and studied their development. And what was pretty clear is that in domestic dogs, you have a dramatic transformation in form with growth, with development. You start off with something that looks like a puppy, has a very distinctive cranial conformation, and gradually this becomes more elongate, like the typical German Shepherd, more wolfy-like dog. Well, you can imagine, and this is what I discovered, if we kind of truncate growth or slow it down, any point in development, we create a skull which is dramatically different. And this is different from the case where you might have proportional growth in a skull, so that if we truncate growth at any one point in our series, the skull is smaller, but it still has the same proportion. So dogs have what we call allometry. That is, they change in shape as they develop postnatally. Other breeds, other species don't have this. And I made this contrast between, say, cats, which the proportions of a kitten are very similar to the proportions of an adult cat. So you imagine if you truncate growth and make a little smaller cat, it's still going to have the same skull shape, but in dogs, you'll come out with a dramatic series of different forms. And that's what I reasoned, this kind of juvenilization, that's what I reasoned is part of the key for understanding diversity in domestic dogs. And this process of kind of juvenilization, we sometimes call it neoteny, is also suggested even in human evolution, that humans uniquely continue to grow their brain at fetal rates after they're born. And to accommodate this expansively growing brain, we maintain these large crania. In fact, a human infant looks more like a juvenile ape than humans do an adult ape. And so this process, maybe in neoteny, might have been important in our own evolution. It's clearly important in dogs. And juvenilization brings us something else too, maybe a behavior that we like. If they're tied, we just truncate growth, we end up with adults that are more puppy-like. And of course, we prefer those playing with our children rather than wolves. So let's talk about the evolutionary framework. When I started out after my PhD, I went to the National Cancer Institute for 5 years, and I learned molecular genetic techniques, and I hoped to find the genes that cause neoteny and all this other shape diversity in dogs, and I set off and soon discovered I had been very naive. You couldn't do it then. And what I did really instead is said, well, what can we do? We can look at the evolutionary history of dogs. And this caused me to go on a several-decade journey into trying to figure out the family tree of the entire dog family, 35 species. And always a little bit unsatisfactory until the dog genome was published in 2005. We used the tools of that dog genome to look at thousands of base pairs and reconstruct this very accurate tree of dogdom and their kin. And for me, this is immensely satisfying, something we couldn't have done when I started at MCI. And it shows essentially in dogs we have three groups, kind of the red foxes, the South American foxes, they have their own little adaptive radiation, and then the dog wolf-like group, the jackals, the coyotes, and African wild dogs. And dogs clearly are part of this group that includes just gray wolves as their direct ancestor and coyotes as the next closest ancestor. That's just one layer, though. That's what exists today. And we have to think of the dog family evolving over many millions of years. And when we look back in the fossil record, we find incredible diversity. We're just looking at the tips of an enormous radiation. Early on, there were dogs that kind of looked weasel-like, smallish, and those turned into more kind of bear-like dogs that ranged in wide diversity in sizes, from raccoon size to very large species. And you can see they're actually the most diverse group in this tree. And just at the very tip are the 35 species of the subfamily Caninae. We can look at another kind of depth across the entire carnivora order, 280 species, all the carnivores we're familiar with. And this is based on DNA data, which shows that dogs are actually the earliest branch in this grouping that includes bears and seals and raccoons and otters. So dogs are the first branch in this part of the carnivore tree, and the other carnivore tree is mostly cat-like carnivores. So what we have today is just kind of a remnant of what existed in the past in the long history of cane day. So just to conclude here, the dog That's ancient divergent from other carnivores, perhaps about 55 million years ago, really at the earliest stages of the mammalian radiation. The extant Canidae, the living Canidae on the other hand, probably radiated about 10 million years ago to all the forms that exist today, so they're a very recent group. And the dog is clearly assigned to the wolf-like group and is very close to gray wolves. All right, so what about the origin and diversification of dogs? And I always try to label everything on slides, because inevitably some student will come up afterwards and say, it wasn't on your slide, it was on the test, and so this will help you out. You won't be tested on this acronym, So the basic questions in dog evolution are really when, where, and how. And the when is important here, because actually dogs are our oldest domesticated species. They're pre-agricultural. They go back to a period where humans were dominantly hunter-gatherers. So they're not like everything else, all other domesticated species that were associated with the agricultural revolution. And we have to actually think a little bit about how this might have happened, where and how it might have happened, because dogs descend from gray wolves, and gray wolves are big. And we want to try to understand what the association was early on that allowed a large carnivore, the only large carnivore ever domesticated, to somehow to be stably incorporated into human society. We know that must be a problem, because we don't leave our children alone with a large wolf-like carnivore. And so there must have been some point of mutualism that developed and selection that allowed for the stable integration of proto-dogs into human societies. And people still think about this today here, Gary Larson. I know you missed the Wainwright's lobby, but they were weak and stupid people, and that's why we have wolves and other large predators. So what we did, and this seems like 1,000 years ago now, in 1997, we looked at some sequences, DNA sequences from the mitochondria, which is actually a little organelle outside the nucleus where the genome is contained, which has roughly about 2.5 billion base pairs in dogs. The mitochondria has its own little genome. It actually evolved from a symbiosis between an early eukaryote and a bacterium. And this functions to produce energy in the cell, but it has its own peculiar little genome that evolves very quickly. That's why we become interested in it. So we looked at a little piece of that genome. It was evolving very quickly. The mitochondrial DNA is maternally inherited, so we're only looking at a female lineage. And we characterized it in a group of dogs and wolves. And this, at that time, was a big sample. So when we walk through the DNA code, we find for gray wolf 1 in its mitochondrial DNA, and here's the sequence, and here are some dogs. And at every position indicated here, there's a difference in these 4 sequences from 4 different individuals representing dogs and gray wolves. And we can kind of tally this difference, and we can compute the average difference. And you can see in dogs, the average sequence divergence between any 2 dog sequences is about 2%, quite a bit. And the average sequence divergence in wolves is about the same, 2%. So that's a lot of variability in the study. In humans, it's more like 3 or 4%. So we can tally all the differences between dog sequences and gray wolf sequences. And here's just, this is not even a political statement, okay? But we can imagine that the sequences of these individuals differ in this way by just counting up the number by which they differ, and we can turn that into a little genetic similarity tree. Now it happens that looks like the Republicans and the Democrats are in different parts of the tree, showing different levels of similarity. We know political affiliation is DNA. I mean, it's based in the DNA, right? This could prove it if it were true. We can do the same thing with wolves and dogs. So it's a little hard to see, but all these are different sequences in a tree of DNA sequences from the mitochondrial DNA. And wolf is with a W and dog is with a D. And we have different groupings. There's this big grouping here that just has dog sequences in it. And there are a number of other groupings that we can define in this tree. And this big one that just has dog sequences in it is a kind of radiation. And we might suggest, well, maybe this grouping, this clade that we might call it, they all have a common origin, indicates the first origin of dogs. The most diverse grouping has most of the dog sequences in it, and whereas other groupings that contain dogs contain dog sequences here, may represent different domestications, like these are just Scandinavian dogs in a wolf. Here's another one where there's some dog sequences, but there's also a wolf sequence that's identical to a dog sequence. That might be recent interbreeding. So this kind of tree suggests interbreeding, maybe separate origins, and diversification. We can use this kind of diversification to go through a kind of dating exercise, because sequence changes are often approximately related to time. So here's this big clade, where we did more sequencing here. And we know from the fossil record that wolves and coyotes diverged about 1 million years ago. They're about 7.5% divergent in their sequence. And if we look at this clade and ask what two sequences are most different, we find that they're about 1% divergent. So 1 over 7.5 times 1 million gives us on the order of like 100,000 years or more. And the point of our paper was this seems a long time ago. Since then, another paper, in fact, one of our collaborators, took a look at this data and more data and said, well, we can kind of juggle the molecular clock a little bit. Seems still to be ancient, but it's more in agreement with aspects of the archaeological record. Well, what is the archaeological record? It turns out dogs have a wonderful and interesting archaeological record. And you might think you know where the first dogs are from, and many of you might say the Middle East, but actually it's from Western Siberia. And they have dogs there that are 15,000 years old. They're clearly dog-looking. Here's a skull, and you can see it has kind of a pushed-in face, upturned snout. And we can measure kind of the length of the palate and the length of the skull here. And here are all wolves, and these are huskies. And here's these Russian dogs. They're actually closer to Great Danes. 15,000 years ago, these dogs are already modified from their close ancestors or close kin with wolves. And there are other remains, too. There are remains, surprisingly, in Western Europe that are 31,000 years old. There are remains in Czechoslovakia, the Czech Republic, which are 26,000 years old. So it seems like actually Europe could be a center of domestication. The other clear center is the Middle East, where we have dogs in close association with humans. In fact, a human with his arm wrapped around a puppy buried that way in the Middle East 11,000 years ago. But that's already agricultural days. These dog-like animals represent probably hunter-gatherer societies that migrated from the spring. So, the next genetic study by my colleague, Peter Savalainen, looked at a much wider sampling of dogs worldwide than we looked at in 1977. He found this intriguing pattern. All these little circles here are different sequences, and how big a circle is proportional to how many of those sequences they found. And what you can see, if you go from East Asia to Europe to Southeast Asia, there are fewer sequences, fewer colored-in circles. And in fact, East Asia has the most diversity of sequences, the most different, or the greatest number of sequences of anywhere in the world. And this is very reminiscent of the argument for our origins in Africa, where diversity is highest. And reasonably, Peter Savalainen suggested, well, East Asia was the place where dogs originated because sequence diversity is highest there. So, here's the story, as was done about a year ago. Dogs have high mitochondrial DNA sequence diversity, and one can interpret that as kind of a diverse origin, maybe from multiple wolf mature lines. Mitochondria is maternally inherited. However, Peter Savalainen found that this high diversity in this one molecule, this one kind of sequence, in East Asia, suggesting dogs may have at least originated there. There may have been more interbreeding with wolves after that. And that dogs are clearly fielded with domestic animals. This is based on the archaeological record and molecular sequence information, 15 to maybe as much as 100,000 years ago. Okay. Then the dog. sequencing genome project began. And every time you want to sequence a genome and have the government try to pay for it, you have to justify it. And you write what's called a white paper, and you say why dogs are unique. And argued in this paper was, I think, a very legitimate argument that dogs are unique. I mean, we want to call all the species we work on, from weevil to whale, model species. And we all make arguments about it. But there is something unusual about dogs, and that is phenotypes, different morphologies, different conformations are replicated in different breeds, like independent experiments. So for instance, this kind of short-legged breed like Corgi and Basset Hound and Dachshund, there are 19 breeds like that have short-legged conformations. There are many breeds which have this kind of pushed-in face, brachycephaly we call it. There are breeds that differ in body size dramatically, large, small. There are breeds that have certain functions, like racing, and breeds which have certain behaviors, such as retrievers. And so those traits are replicated in different breeds, and they give us an experiment. If we march through the genome and we find a difference, say, that is the same in all the short-legged breeds, but none of the other breeds that have normal legs have that unusual variant, we can suggest, well, maybe that variant is the cause of that phenotype, because those 19 short-legged breeds don't share a recent common ancestry. They all come from different stocks. So that's kind of the exercise that I'm going to walk you through. This is a huge effort, sequencing the dog, and it involved, in a sense, two dogs, a boxer named Tasha, and was sequenced at MIT. And Tasha had this unusual property that the whole genome was not very variable, so that made the kind of sequencing and assembly very easy. And another dog, a poodle, which was Craig Venter's poodle, was one of the big founding figures in the Human Genome Project. In fact, Craig Venter had his own sequence, and if you do any kind of manipulations of the human genome world, you're always running into Craig Venter's sequence, and now we have to run into his dog. So these sequences were what we say covered at 7.5x to 1.5x. That just means that any one spot in the genome, we have 7.5 sequences of that spot. We have 7.5 versions of that nucleotide. So that gives us some redundancy, so we can make a really good, greater coverage, the more confident we are in the sequence. And from this sequence, about 20,000 genes were discovered, and that's fewer than humans, which have about 24,000 genes. And we found millions of variants, that is, differences in DNA sequences. And in humans, it's something on the order of 11 million different variants. So that's the totality of the variation in our species. It's probably that much in dogs, too. So this gives us lots of tools to find markers that are variable, and we can study those markers and make conclusions about association. From these variable markers was devised what's called a SNP microarray, and imprinted on that microarray is 127,000 markers. So I told you that most of my life I've been sampling 10 little markers at a time. Here, suddenly, there was a tool where I could sample many orders of magnitude more and say something about how those markers are associated with phenotypes. The cost of the project was $46 million. And that was in 2005. We had to think, well, the human genome cost billions. So actually, this seems like a good deal. So from this kind of resource developed a collaboration association between myself and Elaine Ostrander at NIH, who's a famous dog geneticist, and more analytically oriented person, Carlos Bustamante, and his group, who handled much of the analysis and the useful interpretation of our data. And none of this would have happened unless the maker of the chip, Affymetrix, gave us 1,000 free chips. There was no grant that supported us, really. We just wanted to do it. We tried to get grant support, but the dog never seemed like a good enough model. So here's a canine chip. It has 127,000 markers on it. Some of you may know what this is. This is a floppy disk. is actually floppy, and this holds two standard digital images. And you can't put it in your pocket. You can put this in your pocket. So we participated in this kind of group that went about collecting DNA samples from unrelated dogs, and no dogs were harmed in this collection. In fact, they got a little bit of a tooth cleaning maybe. You just take a little cotton swab and you kind of rub inside their mouth, and that's enough DNA for many of these analyses. And the wolves, unfortunately, don't really stand still for this kind of treatment, and so many of them were sampled after they were anesthetized. And then, this is actually, you can't see it that well, but this is what an array, at least in a cartoon, looks like. The array has little DNA markers bound to it, and we put it on DNA, and where they match, they'll fluoresce. So that marker is there or not, and we know that based on their fluorescence. And it's kind of a photographic detection assay. And this is just as a number of SNPs per million base pairs. We have like 20 or 30 of these markers, what we call SNPs, single nucleotide polymorphisms, every million base pairs. And this is all kind of under the auspices that we had to agree that with Affymetrix to actually make our data publicly available so people can search this database and look at correlations themselves, submit their own data. All right, so let's get to the point now of talking about how we then understood how dogs are built and kind of reverse engineered the dog. We started out with the phenotype and asked what gene changed to make that phenotype. So dogs are formed in a number of ways. One way is by just crossing two dogs that may look alike, maybe from the same breed, and we find that there's a discrete variant of mutant in the litter that results. And humans are great at kind of spying something very different, and then we interbreed that individual as its litter mate, and we fix that phenotype in the offspring. Darwin called these really discrete, different-looking dogs sports. And such dogs were kind of very popular in the Victorian age because there was this push to invent different weird-looking breeds, literally for the nobility, who wanted it as kind of a symbol of their status. And of course, status symbols like that become popular very quickly with the lower classes and everyone else as well. And of course, we breed dogs for other things like strength, and we might pick out mutants in this respect, like the bully whippet, which has what's called a double muscling gene. And individuals which have the double muscling gene and a normal copy of the gene actually run faster than either individuals with two normal copies or individuals with two doubly muscled copies. And you can imagine, we pick breeds for function, but we also pick breeds just for novelty. So there's lots of examples of that. This says really more about the person than it does about the dog. We also cross distinct breeds, and the progeny that results from that cross are then selected in different ways to form new breeds. So we utilize then the diversity of breeds that exist. And lastly, what might be called progressive selection or idea-driven selection, where we have some idea about what breed we want to make, one that herds better, or one that has a certain coat color, or behaves in a certain way. And we select generation after generation for the best traits in that respect. And this is actually what happens, of course, in our domesticated plant and animal varieties. We want corn that grows faster, has more oil, grows in certain environments. We want a cow that's leaner or a chicken that lays more eggs. We do this kind of idea-driven progressive selection over many generations of those individuals. It's difficult to do in dogs, because dogs live a long time, and a breeder eventually dies, and the offspring of that human are probably unlikely to take this up. So this kind of progressive selection is probably not all that common in dogs. Probably what happens most in dogs are these discrete variants or this crossing giving rise to new breeds. Of course, Darwin thought really it was the idea-driven selection. It was the progressive selection that was the best model for what happens in nature. And he used dogs and pigeons extensively in the origin to make the case that artificial selection was like natural selection. But he was focusing on this progressive kind of selection. He traveled all around Britain, interviewed breeders, and tried to understand the secrets of how they created better dogs. All right, so the first phenotype we looked at, of course, was the most dramatic, and that is body size variation in domestic dogs. And actually, this is quite a fundamental parameter in all species, in all animal species, in the sense that it's one of the first things that diverges in irradiation and differentiates animals in different niches. Body size is a very fundamental parameter structure in community. And in dogs, of course, it's very dramatic, from 1 pound teacup poodle to a giant mastiff that's a few hundred pounds or more. So we focused on size to begin with. And this was our procedure, actually. It's called a genome-wide scan. We use all these markers, in this case, the 127,000 chips, to kind of step through the genome of dogs of different size and ask, is there an association of that one spot in the genome with size? And this is kind of a three-part design. We focused first on Portuguese water dogs. And the reason with that is because they have different warts within a single breed. There's a large version and a small version in Portuguese water dogs. So we reasoned, well, if we can find an association in Portuguese water dog between a marker and size, then maybe it's the same gene, the same marker that causes the much wider diversification of body size in domestic dogs. And many of you may know what a Portuguese, who has a Portuguese water dog? Yes, yes, yes. And here he is, interviewing his dogs before selecting one, asking them about their opinions on abortion, gun control, gays in the military, the important issues of the day. What happened with these Portuguese water dogs is that the breeders are wonderful. They volunteered to actually bring their dogs in and let them be x-rayed. So we're able to get detailed measurements of their skeleton and their size, 91 measurements from these x-rays. And 500 of these dogs that had a same pedigree were brought in and this was done. And they were typed, in this case, on 500 markers. So we only had 500, not 127,000. What we found, though, despite this kind of low coverage, was a remarkable association between this one marker, which has this uncomfortable name, which is on Canis dog chromosome 15, was linked closely to this gene called IGF-1, insulin growth factor 1. And this was a great linkage because IGF-1 is responsible for height differences in humans. It also is implicated in a lot of other things like longevity. And so this seemed like a great candidate. So it set us off looking in much more detail in a wider sampling of dogs, from small dogs with their weight in kilograms to large dogs. And what we studied is we marched across the genome near IGF-1. We developed more variants through sequencing. And what you can see here, this is the variability. And it's high to begin with, and then as soon as we get to IGF-1, suddenly the variability drops near zero of our markers. So that indicates there was strong selection there, because the whole region had been swept with fixation in small dogs. Similarly, we can look at the divergence between small and large dogs and march along the genome, and you can see there's a peak in the difference between markers at this IGF-1, as you might expect, because small dogs are selected small size, large dogs are not selected at all. And here's kind of the trait of all our markers. Each little dot is a different marker. These are small dogs in red, and you can see this dip in variability. Here are large dogs in blue, and there's no such dip. So this small size was a feature that was kind of swept to fixation in small dogs especially, and it probably has to do with IGF-1. In fact, we can figure out exactly what particular marker is most closely associated. And when we did that, here's an association analysis. We're just asking what marker is more closely associated with small size in IGF-1. So we can pick that marker and study its properties in a larger dog sample. And when we do that, we find it explains most of the variability. Here's the frequency of this marker, which we call A, allele, and here's body size. And you can see a clear trend. About 50% of the variation in size is explained by this one marker, this one gene. If we did the same operation in a group of you, we'd find the strongest hit probably only explained less than 1% of the variation in size. Similarly, most of what we find in humans and other species are genes of very small effect. In dogs, we have this kind of whopping effect that derives directly from how we built dogs in the first place. We can also study where this kind of gene originated. And one of my students, Melissa Gray, looked at the same sequences in wolves from throughout the world. And what she found is the small gene in small domestic dogs seems to derive from Middle Eastern wolves in this kind of tree. So the smallness seems to have come from Middle Eastern wolves, suggesting that the first real changes in size probably occurred in the Middle East. And we know that's actually true from the archaeological record as well. What about other genes? Our next kind of obvious focus was this foreshortened limb gene. We've seen dogs like this that look like, their bodies need to be shrunk or their legs need to be stretched. And corgis and basset hounds and dachshunds are the obvious examples here. So we compared these so-called achondroplasic foreshortened limb breeds, 19 of them, to a normal breed. And we marched through the genome now with this 127,000 marker chip. And we found one spot where there was almost a perfect association between this marker and these short, foreshortened legs. And why is that spot? Because we had the dog genome, and all the genes have been called or annotated. We knew that there was this gene that was described as a fibroblast growth factor gene. And this growth factor gene has been implicated in the growth of limbs. But what's unique about it is it's a retro gene. That is, there is another fibroblast growth factor gene elsewhere in the genome. Here are all the different chromosomes. There's another copy of that gene elsewhere in the genome. That copy had been transcribed and then reinserted back into the genome elsewhere at chromosome 18. So individuals that have these foreshortened limbs have two copies of that gene. And we know both copies are active because we can actually look at their expression. I don't need this to be complicated, but here's a blob that indicates the expression of this retro gene, FBS4, this extra copy. And these are individuals who are achondroplasia juvenile, and you can see all the juveniles express that gene. They also express the normal copy of the gene. But juveniles that are not achondroplasic, don't have these foreshortened limbs, don't express, you can't see it actually here, don't express the mutant copy. They only express the normal copy, and the gene is not expressed in adults at all. So that makes perfect sense. So we've kind of nailed it here. Similarly, fur types, we can look for associations. Here are all the different dog chromosomes. We looked at smooth coat, wire coat, curly coat, animals from different breeds and this kind of experimental design. Here's a big hit when we compared wild-haired dogs and smooth-coated dogs. So this seems like a gene that may cause smooth coats. In fact, the same gene also seems to cause these facial furnishings in many dog breeds, like I had when I was in graduate school. It's called respondin 2, R-respondin 2. And we can continue to look at hits, and what we found is that just 3 genes, and not important what they are, but just 3 genes can explain 95% of the different kinds of fur types in domestic dogs. And this kind of study hasn't always been done by us. It's been done by numerous groups. We now know coat color genes. We know genes that cause short-facedness. We know genes that cause wrinkly skin. And they all seem to be one dominant mutation. And that mutation kind of spawned around different dog breeds. In fact, for me, it's come full circle. Because I actually used the measurements that I took for my thesis So your thesis never grows old, students out there. But I took my morphologic measurements, and we were able to associate these measurements on skulls and limb bones and teeth with changes in the genome. Here are all the different coded chromosomes, and here are the associations. You can see this gene, this area of the genome on chromosome 5 is associated with all these different measurements of cranial width, zygomatic width, cranial depth, a whole suite of characteristics. The bottom line is here that it's just a handful of genes that explains the diversity in my morphologic measurements that I made on a number of different breeds, champions. So let's try to put this into an evolutionary framework. Let's evolve the dog. And this is work done by Bridget Van Holt, who's in the audience. She's about to finish her PhD in June, move on to a wonderful postdoc at UC Irvine, where she's going to be studying Arabidopsis, the yellow mustard. It's a weed. But actually, it's kind of like the fruit fly of the plant kingdom. Everyone works on it, has multiple genomes. However, I have to admit that no self-respecting dog would pay any attention to Arabidopsis, a weed, even if it was planted near a fire hydrant. It's just but be sure to tease Bridget about this afterwards, because I only have 2 weeks left to do so. So let's look at this tree. This is actually an evolutionary tree. So I just took one of those elongate trees and we circularized it so that it meets at the end. So this is still a tree. It's just made easy because we have all these different breed names. And it's just going to make a lot of sense. Here are all the wolves grouping together from different regions, each region in its own little genetic similarity group. And we can walk through this tree. Here are some very ancient breeds. They're the first breeds to diverge in this tree. They're genetically very divergent, and they include some Asiatic breeds like Chow Chow, Akita, Saluki, Samoyed, Afghan. They're at the earliest point in the dog family tree. They're the earliest divergent. Let's march to the rest of this tree. What we were surprised by is that breeds that just are given functional names like sighthound, seems like it must be a heterogeneous group, seem to be also genetic groups, genealogical groups. There's all these different sighthounds that are designated by breeders. They turn out to be genetic groups as well. And we thought, wow, there must be lots of ways to make a sighthound. You can take a small terrier or a mastiff or a retriever, turn that into a sighthound. Why are they a genetic group? And similarly, when we march to the tree, we found in many cases with herding dogs, with small terriers, with mastiff-like dogs, with scent hounds, with spaniels, that they define a genetic group that matched the functional group, that matched the phenotypic group that was discussed by breeders who don't think about it. So this puzzle, and the explanation, though, seems quite obvious. That is, if you want to make a new herding dog and you're a breeder, you start with other herding dogs, and you cross them. So over time, that breeds a lot of genetic similarity among herding dogs. So you breed like with like, and then select to increase the abilities of your herding dog. But there's something else going on in this tree, too. When we looked at toy dogs, you can see every one of our toy dogs seems to be classified in different functional groups. So this suggested to us, well, if you want to make a small dog, you take one of these very small dogs and you cross it to a larger dog. You shop around this mutation that causes small size. And this, we think, is kind of the dominant axis of how one produces diversity. Take a few discrete mutations, like small body size, like achondroplasia, and you shop that around to different breeds, different genetic backgrounds, and those offspring appear very different because they're on these different genetic backgrounds. So a dwarf mastiff is very different than a dwarf sighthound. And it gives the appearance of diversity where the genetic basis is really quite narrow. And there are other breeds too, that seem heterogeneous like this, the working dogs, and even some of these retrievers. As best we could, we researched the history of dogs that seem to fit into this toy group and asked, well, does the history support this notion? And you can see in many cases indicated in red here it does. That it was crossing between a toy and a herding breed to make a Pekingese. And so this does seem like a dominant mechanism of how one produces diversity. And you can imagine this little bag of genes, this small kit that is then used over and over across all of dogdom that gives rise to this appearance of diversity. And that's the reason, in part, genetically, they're so diverse. Well, there's something else we can use this kind of tree for. Here's a similar tree, just kind of stripped of the long breed labels and laid out flat. And so here are all dogs in these different functional groups. And at the base of the dog tree are wolves from the Middle East. And that kind of suggested to us, well, maybe it's not the Far East, maybe it's not East Asia, maybe it's actually the Middle East that is the origin for dogs, because wolves from there seem closely related to them. So this really wasn't a good enough evidence, and what we did is actually we looked at wolves from the Middle East, from East Asia, from China, and from Europe, and we asked, what of these markers, these thousands of markers that we looked at, is unique to those populations? If I went around after we all left and I swabbed your armrest and recovered DNA from you, in each one of you I'd find something in the order of 40 markers, if I sequenced your whole genome, 40 to 80 markers that was unique to you. And similarly, I'd find another subset of markers that was unique to your genealogy, your family tree. And I'd find another set of markers which was unique to the area where your relatives came from. We can do the same kind of experiment with wolves. We can ask what markers are unique in Middle Eastern wolves, and of dogs, what proportion of those markers is shared with dogs. And that's what this diagram is. There's a Saluki, and this is the proportion of markers unique to Middle Eastern wolves that are shared with Salukis. And you can see across this diagram that in every case, and this indicates significance, in every case, there are more private markers that are always on Middle Eastern wolves, also shared with domestic dogs. And this is kind of going to be a blinding diagram, but I just want to give you the opinion, the feeling that when we look across all dogs, all our breeds, all you see is blue at the top. That is, dogs share these private kind of indicators of ancestry. More of them with Middle Eastern wolves than with other groupings of wolves. And this is a little bit of a different way of parceling the data. And here we begin to see mostly blue, but there's also some black. Europe as well. So Europe is implicated in the ancestry of dogs. And even in some cases, China and Eastern Asia is implicated here. And these are two East Asian breeds, the Chow and the Akita. So here we can tease apart the ancestry of the dogs, and it seems that Middle East is the primary source of genetic diversity in dogs, followed perhaps by Europe, and then lastly, for a subset of dogs, East Asia. We can do more. Or we'll just stop here. We can do more. We can actually look across the genome, comparing dogs and wolves, and ask, where are markers most different between dogs and wolves? And where is there evidence of draw and variability? And this is something that John November and my colleague in our department has done with these data. And what we find is unusual markers that are way out there in the distribution. They have highly divergent between dogs and wolves, and they have really low variability, suggesting selection has been acting. And we can categorize those, and we find basically the top hits, so to speak, seem to have to do with memory and other aspects of behavior, making sense for what happened in the origin of dogs. My favorite hit, though, the one that's most divergent, has very high FSP, very high divergence, is this one. And it's a marker near a gene which causes Williams syndrome in humans. And Williams syndrome is sometimes called the party gene. That is, you walk into a party and there's always someone that's not you, who has a crowd of people around them. And they're talking in a very animated fashion. Everyone likes them. In fact, these people are too friendly, so much so that as adolescents, the real problem that they go up to strangers, they befriend strangers. They're very open and loving and popular because of that reason. And this is kind of a description. This phenotype characterized by elephant facial appearance, low nasal bridge, unusually cheerful demeanor, ease with strangers, coupled with unpredictably negative outbursts, violent outbursts even, and mild retardation, but language skills and a love for music. It's a very unusual syndrome, but it actually matches perfectly to my first dog when I was a kid. A Boston Terrier named Missy. In fact, there's someone in the crowd that's no longer with us. I want to end talking about this very unusual situation in wolves. If you travel around North America and view wolf populations, you find in most all areas black and gray wolves living in the same place. And so this is just the frequency of black in different areas. Where you wouldn't find them so much is in the tundra area, where you just find gray wolves. That makes sense in terms of the camouflage argument, because with the light color background, you think light wolves would be favored there. And these black wolves actually, they gray with age, like humans. They become more gray. And even the gray ones age and become more white with age. Well, we wanted to study this. This bothered us in a sense because it was only happening in North America. In the old world, where wolves come from, there are hardly any black wolves. So we took advantage of the reintroduction to Yellowstone National Park, and this is work done by Bridget Van Holt as well. And we looked at the entire pedigree of the whole population with new genetic techniques. And this is something we couldn't do a few days ago. But with new genetic techniques, we can actually reconstruct the entire family tree. And all the gray individuals, which are kind of gray blobs here, and the black individuals, which are blue, this is their family tree. And we can deduce from that tree that black is dominant, and gray is recessive, and then it's inherited as a single gene, single Mendelian gene. At the same time, co-colored genes were being described in dogs, and we contacted a group at Stanford that had described a new mutation called the K-locus that's involved in melanin deposition in the skin. And we sent them some wolves, and just on a lark, we thought, well, maybe dogs come from wolves, maybe it's the K-locus that's in our population. And sure enough, this group found, now we've done tested many more individuals than that, but there's perfect association between the K-locus mutation that causes black and co-colored wolves. So we found the gene. The unusual thing, though, in this case was the gene didn't evolve in wolves. It evolved first in dogs. And I'm not going to spare you the molecular argument here, but it seems to have evolved in dogs tens of thousands of years ago. And then at some later time, it was transferred to wolves, uniquely in North America. And we only find black wolves in the old world where actually there are lots of dogs. And that this probably occurred some time ago, that is, it wasn't the dogs living here today, it was dogs brought here by Native Americans. They were probably much more loosely associated with humans. That's when the black transfer took place, and that coat color was swept through North America. So it does something, we don't know quite what. And that's the essential question. Well, we learned from this that actually genetic variants that form under domestication or through genetic engineering can be successful in the wild and actually transform species. What it does, we can't be sure, but we've looked at Yellowstone. And this is work done by another student in my lab, Dan Taylor, who is an ecologist. And he's looked at the longevity, and this is age in years and the probability of survival, for black and gray wolves. And black wolves live longer than gray wolves, a year and a half longer on average. And these gray wolves only live 6 or 7 years. And we still don't know why this is the case, but the K-locus is actually in a class of genes called defensins, and they are involved in immunity. So it may have something to do with immunity to disease, which is the principal cause of devastating mortality in Yellowstone National Park. This is something that we're working on now. So let me just conclude here. The adaptive radiation of dogs is involved with a relatively small number of genes with large effect. This is very different than humans or any of our domesticated plant and animal species, where there's many genes, a small effect, just a few percent at a time. So we were able to find these genes in dogs because they're such large effects. These variants can be transferred to different breeds and different genetic backgrounds by crossing, forming the kind of appearance of a lot of diversity because they're different genetic and phenotypic backgrounds. And that's just actually a mutation as well as this kind of goal-driven selection limits diversity in dogs, where it kind of depends upon mutations there and here, and that mutation has to be very discrete. Variant selected under domestication can be transferred to the wild and affect fitness in wild kin. And I'm just going to end telling you what we're doing now. This is a wolf genome project, our first genome. We've arranged some tentative support just to initiate this project. In fact, probably next week we'll start the whole process of the wolf genome, but we don't know how much more we're going to be able to do this. And this is a student in my lab right in Schweitzer. You can see a two-type like that. You can see, and John Novembre, who's actually heading this project. But this is a very exciting time to us. Just like the Neanderthal genome that was described last week in Science is so fundamental to understanding what makes us human. I mean, that Neanderthal genome discovered certain genes that only change in modern humans. And many of those genes seem to have to do actually with autism. So that is when they misfunction. And the notion is that maybe what's unique about humans is that our ability to engage, to be highly social, where most other species of mammals are more on the autistic side of things. But it tells us really what genes make us human. In the same way, we can look at wolves, which are even closer to dogs than Neanderthals were. And we can ask what differences are there in the genome and understand actually what makes a dog the wonderful animal that we all enjoy so much today. So I just want to acknowledge that there's, of course, a whole team of collaborators involved at UCLA and NHGRI, NIH, and Cornell University. And then, of course, I would be remiss if I didn't mention my lab as well, and the wonderful support group that has brought us to all this work over the years. Thank you very much.